Let's start. Okay, so now you do have action potential. Now you do have action potential coming down from the axon, right? All of the sodium getting in. Am I making some sense? And then of the potassium getting out. And then you have action potential. So what? What happens next? Okay, what happens next inside of the axon knob? You remember we already talked about them. I showed them to you guys, everything. At the end of the axon knob, right here in the knob, you do have neurotransmitters. In Bio 1, I did talk about it, but I don't know, it depends on the time that the professor you had. These vesicles are formed, and when the vesicles inside of a neurotransmitter, they're gonna get on a railroad track, and they get out of the axon terminal or axon knob. So this is made up of, all of these are made up of phospholipid. So that phospholipid attaches to the cell membrane phospholipid, then they diffuse, they release the neurotransmitters to the next neuron. Am I making some sense? So the function of the action potential is to release the neurotransmitters at the end, eventually. Release neurotransmitters, okay? And then those neurotransmitters go in a synapse. A synapse is space between one neuron and another neuron. That's why they call this neuron, that neuron, they call it presynaptic neuron. They call this neuron right here, that neuron, they call it postsynaptic neuron. So these are all axon terminals or axon knob of presynaptic neuron, and this is postsynaptic neuron. Right? So you all know what synapse means. Synapse, it means space. The junction, if you would, that space is so small, very small space, but there is a space. The junction, the space between two neurons is called synapse. Okay, so when the neurotransmitters are released to the next neuron, what do? I stimulated this neuron, right? I didn't stimulate this neuron. Am I making some sense? When the stimulus arrives, it arrives to that neuron. So that neuron takes the action potential and releases it to this neuron by chemicals, these chemicals called neurotransmitters. And those neurotransmitters go and open up, they cause the sodium channels to open up. When the sodium channels open up, what will happen? Sodium rushes in, and now you have action potential on postsynaptic neuron. That's it. I rest my case. I'm done. Am I making some sense a little bit? So this neuron is releasing neurotransmitters to that neuron. That neuron is not being stimulated physically, it's being stimulated chemically. Release of neurotransmitters. The most common neurotransmitters in human body is acetylcholine, which I guess I'll come back to it. And here they are showing you a little bit of, these are the sodium channel, they cause the sodium channels to open up. They open up and sodium comes in, and you have action potential in postsynaptic neuron. Here again, another picture of it. Here again, another picture of it. One thing I'm forgetting to mention, very, very important, for these these vesicles. This is the mitochondria, right? You remember that. These vesicles to be released, you need calcium. Calcium are going to diffuse from outside of presynaptic neuron. This is a presynaptic neuron, this is postsynaptic neuron. So calcium is going to cause, the calcium causes these vesicles to move. It's needed for their movement. In Biology 1, we studied that a little bit, and then they come and of course release the neurotransmitters. So calcium is needed for release of vesicles. I hope I'm making some sense. Yes? Okay, here again, same pictures. I'm going over and over and over. This is a nice picture. I got it from somewhere. I think, is this in your textbook? I don't know if this is in your textbook. I got it from somewhere else. So this pretty much puts everything into perspective, what you should know. And then it gives you the name of the neurotransmitter as well. Acetylcholine. There is an enzyme on postsynaptic neuron called acetylcholinesterase. Acetylcholinesterase. ACE at the end of the word in biology means what? Bio one. ASE at the end of the word means what? Enzymes. Thank you. Enzymes. So this is an enzyme right here that breaks up acetylcholine. Of course, some of them are being used. Some of them are being, there's so much release. Some of them are being destroyed, and then they go back to presynaptic neuron. Acetyl and choline, they both go back to presynaptic neuron to go the cycle again and again and again. So this is it. I'm not going to read it to you guys, but this is pretty much putting everything together. If I ask an essay question, this is possibly question number 7, 8, 9, what happens between presynaptic neuron and postsynaptic neuron? Right? Everything you need to know is in that slide. What happens? What happens? Yes, that's a neurotransmitter. In this class, you only have to know, maybe I think, you only have to know 2 or 3 neurotransmitters. There's a long list of them. There are a few of them, but you only have to know 2 or 3 of them, which I will mention later on. Okay, this is the one that you should know. Acetylcholine is a neurotransmitter made up of acetyl and choline. Okay, so let's move on. Here is it. I said a neuron might end up in another neuron, right? That's one place neuron end up, in another neuron. However, neurons can end up into muscle. Okay? So a neuron can stimulate a muscle, a neuron can stimulate another neuron, and neuron, the third place is, a neuron can stimulate what? The third one. You're taking YouTube time.
The brain?
No. Gland. Is that right? Stimulate your glands too. The neurons can stimulate your glands. Glands of the body. Some of your neurons are being stimulated by, some of the glands are stimulated by neurons. So 3 things. Neurons stimulate 3 things: muscle, glands, and another neuron. So we are going to talk about what happens in the muscle when a neuron is being stimulated. So you have all of the components right here, and this is the slide you have in the lab. This is one of your microscopic slides right there. I should turn off more lights, but I'm not going to. This is all muscle. These are all skeletal muscle. This is a nerve bundle. And the nerve bundle comes a branch, and branch. This is your axon. And then these are your axon knobs. And then, of course, when you stimulate it, then the muscles contract. And I will talk about the muscle contraction later. So this is pretty much same as that one. But in this one, you have so many of them. In here, they just teased, and they did draw for you only one. Okay? So let's talk about an application of it, an application of what we learned so far. If you go to a doctor and they try to check your reflex, right? I don't know, you sit on the doctor's those examination tables, and they check your reflex. So this is what happens. A neuron is called a sensory neuron, right here. It takes that that stimulus, right? The stimulus I gave him this morning is like this, but the stimulus that you go to the doctor is a light. They want to check your reflex. Is that right? Or they put the hammer right in your knee. Okay, and then it goes from sensory neuron to another neuron in spinal cord. We do have a lot of spinal cord. It's called association neuron, and from association neuron it goes to motor neuron, and motor neuron end up in your skeletal muscle, and your skeletal muscle causes your leg to jerk. So he knows your reflexes are fine. And this. The same thing works with the eye, and we'll talk about the eye too later on when we get to the eye. So this would be a presynaptic neuron. The sensory neuron is a presynaptic neuron, and then the postsynaptic neuron would be a motor neuron, or association, or interneuron, or another name for interneuron is called association neuron, right here. So presynaptic neuron, sensory neuron from the skin, to when you touch something hot, you quickly remove your hand. Or you touch something sharp, you quickly remove your hand, right? Same thing, it's called reflex arc. So, right here you're touching something sharp, the sensory neuron goes to interneuron, and interneuron is postsynaptic neuron. Now, after this goes back to resting potential, the sensory neuron goes to resting potential, you have action potential on this one. This one becomes presynaptic neuron, and motor neuron becomes your postsynaptic neuron. So they're just sort of relating. Have you guys have you ran relay, or swimming relay, or running relay, right? When I pass the baton to the next one, I'm not in action anymore, right? You all know what I'm talking about, relay? Track and field relay? Right? I pass the relay, I'm done. I'm kaput. Shish kebab. Right here. It's this guy who's going to run, and he's going to become presynaptic neuron. And then, of course, he's waiting until he gets neurotransmitters. You all know what I'm talking about. Neurotransmitters are released from here to here. Neurotransmitters from, or the action potential goes from here to here. Neurotransmitters are released from here to here. And then you have action potential, it goes, and neurotransmitters are released from here to muscle, which I will talk about that next, not today. And then we'll talk about what happens in the muscle. You should read ahead, you should watch the videos ahead, you should look at the slides ahead. Okay? Anyhow. So, and then of course your brain, you know, you have your brain and spinal cord. I will talk about brain in a little bit. Your brain, your nervous system kind of counteract each other when one, your sympathetic system and parasympathetic system. Do not worry about this too much. I will not ask too many questions, but I thought maybe I should put this up if you want to study it for your own. But that slide, you do not have to worry about it. You have, our nervous system is broken down to two parts. Your central nervous system, which is your brain and the spinal cord, and then you have peripheral nervous system, periphery of your body. The rest of your body is peripheral nervous system. The central nervous system usually is broken down to parasympathetic and sympathetic nervous system. And for example, one neuron dilates the eye, the other one neuron constricts the eye. Okay, so they work against each other. So you live in a homeostasis. You live in a balance. If your eye are dilated so much, you eventually become blind and the person dies, even for dilation of the eye. I'm exaggerating a little bit, but anyway. So that's why they work opposite of each other to slow down, in most cases, to keep body in a balance, Okay. Parts of the brain you should know. I'm expecting you to know the lobes of the brain. You have the frontal lobe, you have your parietal lobe, you have your occipital lobe, temporal lobe. Those are four lobes, L-O-B-E. Those are four lobes of the brain. So those are the four lobes you should know. On the model we have in there, so I put a piece of tape, a color tape in the lab practical exam. I say, what is the name of structure? Number 5. And then you have occipital, temporal, frontal, parietal, occipital. Did I say occipital? Yeah, occipital in the back. Okay, and then the other parts of the brain, the cerebrum, those four lobes make up cerebrum. And then you have cerebellum. Cerebellum looks like a cow dung. Have you guys seen cow poop? Layer. Look like that, cerebellum. So that's another part you should know about cerebellum. And then you have your pons. I would expect you to know the pons. And then your medulla oblongata, which is the extension of spinal cord. Okay? Connects, medulla oblongata connects brain to spinal cord. So brain and spinal cord are connected together. As far as the function goes, yes, whatever is on, not all of these details, but there is, I thought there's, yes, I think it's this one. Maybe it's that one. There's not much that I expect you to know. Maybe I should have made you a table that what you should know, but definitely, you know, occipital is involved in your vision, okay, and then your parietal, your motor and sensory parts, do not worry about all of these, are your parietal lobe, and then your frontal lobe, speech, smell, okay, involved with lobes, again, and your temporal lobe, your hearing, okay, so those are pretty much what I expect you to know, smell, speech, frontal lobe, your temporal lobe. It's more than that, but again, why I'm asking you to know this, you're learning the basics, the very, very basics. And then later on, when you go to the next course, they build up on your basics. They're assuming you already know the basics. Okay, so on the quiz on Tuesday, I will ask definitely, what is the function of occipital lobe, vision, or put it in the matching situation. So you should know that. So parietal is motor and sensory, and that's about it. Speech, and then pons, and cerebellum, definitely know something about cerebellum, balance and your coordination. It's all part of cerebellum. Okay, and then of course we can talk about brain for the whole entire semester, but you know, we have to move on. Maybe, well, let's move on. The eye, the only sense organ that we're going to talk about, there are 5 of them, is the eye. Of course, we talk about skin, but I'm not going to talk about the nervous system of skin. Just talk about physiology of it, and that's about it. Anatomy of it, sorry. Histology of it, and that's about it. But eye, these are the parts of the eye that you should know, and their functions. Parts of eye and their functions. So the very outer layer of the eye is your cornea, right here, which is transparent, clear portion of your eye, and the most successful transplant are cornea, because there's not much blood vessel in there. They take my cornea and put it on your eye, you will not reject it. Most 99% success, maybe 100% success. And after cornea, you do have the iris, which is the color. Everybody's iris color is different, and that's a muscular portion. When you go to a doctor, they reflex, they put a flashlight in your eye, they're checking your reflex. When there's too much light going in here, too much light, it will close. At night, when it's dark and you're coming to a room, this opens up extremely. Okay, so the doctors can look at it and see what is your reflex by closing the iris and opening up the iris. And behind the iris is the lens. I'm going over the parts that you should know. And ciliary muscles, these are the ciliary muscles. When you have an object far or near, they cause the lens to adapt itself to the distance of the object from your eye. Then you have
pupil or iris.
Oh, pupil, I'm sorry. Pupil is the opening in the iris. Thank you for bringing that. Okay, pupil is the opening in the iris. So they look at your iris, they close and open. When there's too much light, the iris close, so the pupil does not allow too much light to go through. So there's not much light going through pupil. Pupil is just the opening of the iris. Okay, and there is another fluid chamber in here too. Your textbook didn't mention, I'm not going to mention it again. This one is called inner chamber, vitreous humor. All of that fluid inside of the eye is called vitreous humor. Okay, then in the back of the eye, I forgot to mention sclera. Yes, sclera is the white portion of your eye. It's made up of connective tissue. Very, very, very tough to cut into. It's almost as hard as your bones. Very tough connective tissue to cut into. That's the sclera. That's a white portion of your eye. And you cannot see anything through your white portion of your eye. Okay, I hope I said everything. In the back of your eye, you have retina. Retina, that is where the image is formed. That's where you have rods and cones. Rods and cones are responsible for your vision. Rods are responsible for seeing in black and white at night, and cones are responsible for your color vision. We have 3 different types of cone: blue, green, and red. Blue, green, red. Those are 3 different types of cones. And with combination of those, then you can see yellow, you can see other colors.
I thought it was yellow.
Blue, green, red. Those are the 3 different types of cones you have in your eye. Okay, and then behind retina, you have choroid. Choroid is your muscular portion of your, is a vascular, your blood vessels. There is a whole bunch of blood vessels on the choroid. Whole bunch of blood vessels on the choroid. And then of course, this is your nerve, optic nerve right here that takes the image that is formed in the back of your eye on the retina. It takes it to the brain, and of course in the brain it makes the image upside down. Makes it here you see the image upside down. Right now in your retina you're seeing my head here and my feet here. Of course you cannot see, but your brain will fix it for you. When it goes, the image goes to the brain, and the brain put my feet on the bottom and my head on top. But then, and then you have fovea centralis. Fovea centralis is accumulation of a lot of where there is a lot of cones. Where there is a lot of cones, that's what you call your fovea. And we do have all of these parts are on the model. Are on the model. All of those parts are on the model. You have any question during the exam, during the lab, ask me. Okay, Amir, where is vitreous humor? Amir, where is the lens? Where is iris? Where is retina? Retina is the part that is not very clear, but I can show you. But anyhow, optic nerve and so on. And then, of course, image is not formed here. Optic nerve is your blind spot. Somebody asked what is blind spot? That's your optic nerve, right? And here they are, rods and cones. And that's all I want you to know. Don't worry about the rest of them. Let's start right here. I'm going to start on next chapter, and hopefully I have these 2